Perfect. We start sharing my screen just to sum up what is the Young Investigator initiative. It's a way to attract young people and to level up the skills of young trainees in neuromuscular conditions. We hope to attract through a number of seminars related to clinical, MRI, genetics, histology tools to attract young clinicians, biologists, and people who are supposed to be the next generation of neuromuscular disease experts. Please, if you are interested in looking the previous webinar record, register yourself with the QR code. That said, just these are the agenda. We have today electronic genetic data and clinical report form. So we have the genetic part of the Young Investigator Training Initiative. Then we will have in the spring an additional webinar on the CoMPaSS-NMD Atlas. And finally, we will have in the summer school, where we will wrap up what has been done in CoMPaSS. With this in mind, I will ask the coordinator of the CoMPaSS-NMD, Rossella Tupler, to present the philosophy behind the project.
Thank you very much, Filippo. Thank you to all participants. I am very glad and proud to be introduced the fourth webinar on CoMPaSS. What is very important, we started, of course, about what we don't know. How can we reduce this odyssey that the patient can undergo to, simply because we know little, we don't know that much. And CoMPaSS is really trying to learn from past data and from our experience in order to get through the most advanced tool generated by machine learning and artificial intelligence to create something that can improve our ability to make a diagnosis and support patient and support knowledge. What is very important what I think it is very important in CoMPaSS is interdisciplinarities. And I think that we are seeing this happening because people with different background, neurologist, bioinformatician, computational scientists, or molecular biologists are trying to talk and try to convey needs and ideas. So this is really the reason why we are doing this for young investigator. And I am thanking you, and I hope you will be able, or you will be willing to participate to the next occasions. And if you want to contact us, please feel free. And if you want to be involved, please feel free. And if you have any question, please feel free. Thank you very much.
Thank you, Rossella. This is just an introduction to CoMPaSS. Now I will ask Annalisa I have to see how. Okay, great. We will have the first presentation from Giorgio Codullo from CGAT, SME based in Germany, I guess Tübingen. And then Giorgio will give us a genetic overview. Unfortunately, Giorgio is not with us today, but he was so kind to record this presentation and to allow us to see details on the new kind of genetics we can apply into CoMPaSS. That said, I will move. I can't hear. Annalisa,
Me
perhaps.
too.
Yeah, perhaps there is some technical problem.
Yes, I try to reach. Can you see the presentation?
Yes, we see the first slide.
Okay.
Good morning, everyone, and thank you for joining this session.
Can you hear the voice?
Yes.
Okay.
Today, I will give you an overview of the sequencing technologies that underpin modern genomic diagnostics, with a particular focus on neuromuscular diseases, NMD, and the goals of the CoMPaSS-NMD project. Over the past 2 decades, DNA sequencing has become a cornerstone of rare disease diagnostics. It plays a key role in understanding, classifying, and ultimately diagnosing conditions that would otherwise remain undefined. This is especially true for neuromuscular diseases. As for today, many patients still do not receive a clear genetic diagnosis. The CoMPaSS-NMD project aims to address this challenge by combining deep clinical expertise with cutting-edge genomic technologies and advanced informatics. Our goal is to improve diagnostic outcomes for patients with NMD by using the most appropriate and effective tools available. So, in this presentation, I will walk you through the clinical rationale for sequencing, the types of genetic change we aim to detect, and how different sequencing platforms contribute to that effort. Before we move forward into the discussion on sequencing, it's worth taking a brief moment to review a fundamental concept, the central dogma of molecular biology. While we could spend an entire lecture on it, here is the essence. Genetic information flows from DNA to RNA to protein. Understanding this flow is critical for grasping the basis of medical genetics. If a mutation occurs at the DNA level, it affects RNA and ultimately leads to the production of a dysfunctional or missing protein, and that's where diseases begin. Let's now look at the genetic basis of neuromuscular disorders. In most cases, over 80%, these are monogenic conditions, meaning a mutation in a single gene is responsible. But despite this apparent simplicity, we often observe a high degree of clinical and genetic heterogeneity. This means that patients with similar symptoms may have mutations in different genes, and conversely, mutations in the same gene can lead to very different phenotypes. In short, the same cause can result in different outcomes, and the same outcome can stem from different causes. This complexity also impacts treatment strategies. In some cases, even family history may not help, especially when we're dealing with de novo mutations, which are not inherited from either parent. The type of mutations involved can range from large structural changes, such as deletions or duplications, to repeat expansions or copy number variations. Variants affecting the genome can be broadly classified into 2 main categories. The first includes sequence-level changes, like SNPs, insertions, or deletions, which alter the nucleotide sequence of a gene. The second group includes structural or length-based variations, such as deletions, duplications, inversions, or translocations of large genomic segments or entire genes. These are referred to as structural variants, SVs, and within this group, we also find copy number variations, CNVs. Structural variants, especially the CNVs, and repeat expansions are among the most common contributors to the pathogenesis of neuromuscular disorders. Although this concept may already be familiar, it's important to reiterate the genetic nature of NMDs before we dive into sequencing strategies. As mentioned, the vast majority, about 80%, of NMDs are monogenic, so caused by a mutation in a single gene again. These typically follow Mendelian inheritance and are best investigated using targeted approaches, such as single gene testing, gene panels, or broader sequencing like WES or WGS. However, there are also polygenic forms, such as certain sporadic cases of ALS, where the classical sequencing approach may not be sufficient or informative. Understanding whether a condition is monogenic or polygenic is therefore crucial, as it directly guides the diagnostic strategy we choose. In this slide and the next, I've simply tried to visually reinforce something we've already discussed in details, the concept of heterogeneity, both genetic and clinical, that characterize the NMDs. Let's now move to the molecular techniques used to study nucleic acids, particularly focusing on how these methods evolved into what we now know as third-generation sequencing. Let's start with the basics. I assume most of you are already familiar with polymerase chain reaction, or PCR. PCR allows us to take even a tiny amount of DNA, called template, and generate millions of identical and exponential. Each cycle doubles the target DNA fragment until the regions are exhausted. But it's worth noting that PCR is limited to amplifying fragments up to about 500 base pairs in length. Think about what it means. To amplify or sequence an entire human gene, which can be several kilobases long, you'd need multiple rounds of PCR with different primers. In fact, the Human Genome Project began this way, sequencing the genome fragment by fragment using PCR. Fascinating, isn't it? But clearly time-consuming and not scalable for large-scale diagnostics. Before the advent of high-throughput sequencing, Sanger sequencing was the gold standard for DNA analysis, and it's still used today for specific clinical applications. This method is based on the same principle as PCR, but with one key difference. It uses modified nucleotides called dideoxynucleotides, or DDNTPs, which terminate DNA synthesis at a specific point. As the DNA is replicated, these DDNTPs are randomly incorporated, generating fragments of varying length, each ending with a fluorescent-labeled base. By separating these fragments by sides, typically via capillary electrophoresis, we can reconstruct the exact nucleotide sequence. However, Sanger sequencing is much more time-consuming and labor-intensive than NGS method. It typically allows for sequencing of around 500 to 900 base pairs per reaction, which makes it unsuitable for large-scale or genome-wide analysis. That's why Sanger is now used mainly for confirmation testing or very targeted sequencing, while broader diagnostics rely on next-generation sequencing technologies. The Human Genome Project played a major role in advancing sequencing technologies. What once required a month to sequence can now be done in just a couple of days, even for a whole genome, thanks to next-generation sequencing, or NGS. Unlike Sanger sequencing, which analyzes one fragment at a time, NGS works with sequencing millions of fragments in parallel. DNA is first fragmented, adapters are ligated, and the library is amplified. Each fragment is then read through cyclic reaction, producing millions of short sequences that are later aligned and assembled computationally. Different platform uses different chemistries and detection methods, but the core principle is the same: massive parallelization, which dramatically increases throughput. This makes NGS ideal for large-scale applications like gene panels, exome sequencing, or whole genome sequencing, and is the current standard in most genetic diagnostic workflows. Illumina sequencing technology is based on a principle known as sequencing by synthesis, or SBS. But to understand it clearly, we should think of it as a highly miniaturized and massively parallel version of PCR and Sanger sequencing together. The process starts with fragmentation of the DNA we want to sequence and ligation of adapters to both ends of each fragment. These adapters are short synthetic DNA sequences that allow the fragment to bind to the flow cell surface, which acts as the stationary phase in the system, and to be amplified and recognized during sequencing. The flow cell is essentially a glass slide coated with oligonucleotides that are complementary to the adapters. DNA fragments bind to these oligos and then undergo bridge amplification, a form of in situ PCR where each fragment is copied multiple times to create a dense cluster of identical sequences. This step is essential, cluster formation, because the sequencing signal from a single molecule would be too weak. The cluster boosts the signal and makes it detectable by the optical system of the sequencer. During sequencing, fluorescently labeled nucleotides are incorporated one at a time. After each cycle, the instrument captures an image of the flow cell and records the fluorescent signal from each cluster. Since this happens in millions of clusters at once, the system can read a vast amount of DNA in parallel, with read lengths typically between 100 and 300 base pairs. The final steps includes base calling, alignment to the reference genome, variant calling. In CoMPaSS-NMD, we use Illumina-based short-read technology for both whole genome sequencing and for our, from CGAP, exome extra diagnostic analysis. We talk about it more later. While it doesn't capture long structural changes, the short-read sequencing, it remains a highly accurate and efficient method for detecting point mutations, small indels, and CMVs across coding regions. Let's now move into the realm of third-generation sequencing, or TGS. Unlike NGS, which relies on short-read amplified fragments, TGS technologies can read single DNA molecules in real time, often without PCR amplification, and with read lengths in the tens of kilobases. One of the main TGS platforms is the PacBio, which uses a method called SMRT, Single Molecule Real-Time Sequencing, or SMART. In the system, a single DNA molecule is immobilized in a tiny well called zero-mode waveguide, where DNA polymerase incorporates fluorescently labeled nucleotides. The incorporation is monitored in real time, allowing direct observation of the synthesis process. PacBio produces what are known as hi-fi grids, which are long, typically around 10 and 25 kilobases, and highly accurate. Compared to short-read technologies, SMART sequencing excels in detecting structural variants such as large insertions, deletions, repeat expansions, and complex rearrangements, all of which are relevant in neuromuscular disorders. The trade-off is a lower throughput and a higher cost per sample, at least as of today. That said, we hope that in a future phase of the CoMPaSS project, we will be able to take advantage of long-read sequencing technologies, which we in CGAT already provide as a service, to further improve diagnostic resolution of selected cases. Another third-generation sequencing technology, which is gaining more and more success every day, is Oxford Nanopore, which uses a fundamentally different principle. Instead of synthesizing DNA and detecting fluorescent signals, nanopore sequencing detects electrical changes as a single DNA or RNA strand passes through a nanopore embedded in a membrane. Each base alters the electric current in a specific way, allowing the system to infer the sequence in real time. The advantages of this technology are that It does not require PCR at all, does not use fluorescent dyes, and can produce ultra-long reads ranging from few kilobases to even more than 1 megabases in length. The trade-off is that base calling is more complex. Accuracy is lower compared to Illumina or PacBio. Typically, raw read accuracy ranges from Q10 to Q20, although this is improving with new chemistries, duplex strategies, and advanced base calling algorithms. Oxford Nanopore is particularly suited for very long, repetitive, or structurally complex regions, but it also requires highly specialized analysis. In the next slides, we will dive into the advantages, limitation, and specific application of each method in the context of NMDs. Whole exome sequencing, or WES, targets only the protein coding region of the genome, so the exons, which represents just 1 to 2% of the genome. However, and this is crucial, over 85% of the known disease-causing mutations are located in these regions. That's why WES remains one of the most effective and widely adopted tools in clinical genetics. It allows for comprehensive analysis of coding regions while producing a smaller and more manageable dataset compared to the whole genome sequencing. It is also more cost-effective, which makes it suitable for routine diagnostics. That said, WES has some limitation. It is restricted to exons and therefore misses variants in non-coding, deep intronic, or regulatory regions. The coverage can be uneven, and some genomic targets resist to enrichment, leading to incomplete capture. Additionally, structural variants like CMVs and large rearrangements are more difficult to detect with this approach, unless they fall within well-covered exonic region. Still, for monogenic diseases, including many neuromuscular conditions, WES remains a powerful first-line sequencing tool. When WES limitations are a concern, or when WES fails to provide answers, we move to whole genome sequencing, or WGS. It allows us to sequence the entire genome, coding, non-coding, intronic, and intergenic regions. It does not require enrichment or capture, which makes it less biased and more uniform in coverage across regions. This approach is crucial when we suspect involvement of regulatory regions, splice sites, deep intronic mutations, or when we want to detect complex structural variants and repeat expansions that may fall outside the exome. Additionally, WGS is better at identifying non-coding pathogenic variants or regulatory mutations that would otherwise be missed, and allows for genome-wide CMV detections with higher resolution and breakpoint precision. Of course, WGS has its own trade-offs. It is more expensive, requires larger-scale data storage and interpretation, and typically delivers a lower depth of coverage per base than targeted approaches. It stands around 30x against 100x of the targeted approaches minimum. However, in the context of neuromuscular diseases, which often involve structural complexity, genetic heterogeneity, and sometimes unusual variance locations, WGS represents the most unbiased and comprehensive tool we currently have. To overcome the main limitation of both standard WES and WGS at Seagate, we developed the Exome Extra, combining the targeted power of exome sequencing with features typically associated with genome-wide analysis. While WES is efficient but limited to canonical exons, and WGS is comprehensive but resource-intensive, Exome Extra offers enhanced clinical resolution without the overhead of whole genome sequencing. This assay extends coverage well beyond the standard exome. It includes UTRs, clinically relevant deep intronic regions, and splice sites, areas frequently involved in pathogenic mechanisms but often missed by conventional exome designs. It is also optimized for diagnostics use, offering greater uniformity of coverage, reducing dropouts, and improving variant calling accuracy across critical regions. Furthermore, a key feature of Exome Extra is the inclusion of a genome-wide CMV backbone, which enables detection of copy number variants across the entire genome, not just within targeted exomes. A significant advantage over both WES and WGS in terms of CMV resolution versus cost. In essence, Exome Extra brings together the efficiency and interpretability of targeted sequencing with much of the genomic breadth and flexibility of WGS, making it a powerful tool for diagnostics, particularly in heterogeneous disease such as NMDs. Now that we've reviewed the main sequencing technologies, we can look at how they are applied in practice, specifically in the diagnostic path for neuromuscular diseases. In the next slide, we will explore when and why we moved from a single gene test to a broader strategy like WGS, what limitations still exist in short-read sequencing, and how we can best navigate this challenge to improve diagnostic yield in NMDs. Given all the above, it now shall be clear that the selection of the appropriate sequencing strategy in neuromuscular disorder depends on the clinical context and the level of suspicion. When there is a clear clinical diagnosis pointing toward a specific gene, a single gene test, often by Sanger sequencing, can be appropriate and cost-effective. However, in most real-world situations, clinical presentation is variable and overlapping, and we often lack of precise gene hypothesis. In those cases, we move to a multi-gene panels or to a broader approach such as WES or WGS. These comparative tables highlights the main differences. While target panels have high depth and lower costs, WES and WGS offer greater diagnostic power, especially in complex or undiagnosed cases. Next-generation sequencing, particularly short-read platforms, revolutionized the diagnosis of NMDs, uncovering hundreds of causative genes and new mechanisms. But despite these advantages, diagnostic yield remains incomplete. Many patients still receive negative or inconclusive results. Several categories of pathogenic variations tend to escape short-read detections. These include structural variants, the biggest one, repeat expansions, non-coding regulatory variants, variants of uncertain significance or VUS, and disease with a complex or polygenic mode of inheritance. Addressing these gaps often requires integrating additional techniques, including long-read sequencing, transcriptomics, MRI imaging, functional assays, and others. Finally, in practice, genetic testing for NMDs follows a stepwise logic. If there's a strong clinical suspicion toward a known gene, such as DMD, SMN1, or PMP22, a single gene test can be a starting point. If this yields no results, or if the phenotype is less specific, we move to multi-gene panels, covering the most relevant NMD genes. When even panels are not sufficient, or when structural variants or unusual inheritance are suspected, we escalate to WES or WGS. The flowchart in the slides shows how decisions are made in relation to clinical probability and diagnostic breadth. At each step, we must remember that short-read sequencing still has limitations, especially in resolving genome structure, identifying deep or regulatory mutations, or interpreting complex variant combinations. That's why the future of NMD diagnostics likely lies on combining sequencing approaches with improved bioinformatics and multi-omics strategies. That brings us to the end of this presentation. I'd like to thank you all for your attention. I hope the information shared today has been useful, not so boring, I hope, and I'm of course happy to continue the conversation, to answer questions or dive deeper into any of the topics we've touched on via email, so write me anytime. Thank you again and enjoy.
Thank you, Giorgio. It was very nice presentation, a sort of recap of the genetics in tools and how they can be applied to neuromuscular disorders. I guess today Giorgio is not attending the webinar, but as he said, he will be happy to reply to your questions. So you can use the chat to write your question. We will take notes and send to Giorgio. But meanwhile, we go ahead, and it's my pleasure to introduce Anna Toff, who is the Senior Research Associate at the John Walton Muscular Dystrophy Research Center in Newcastle, in the UK. Anna is a geneticist by training, and she got her master in molecular genetics and PhD, if I'm not wrong, in biological sciences in the UK. She is masterable as a myologist broadly. She will present how we apply genetics to CoMPaSS and electronic form for genetics she has contributed to design for CoMPaSS. And I guess you have the ability, you can use your slides. Yes, thank you. The stage is yours.
Thank you very much. Filippo, I don't know if you can see my slides now.
Yes.
Not yet? Yes? Okay,
yes.
Thank you very much. Well, I should thank also Giorgio for that brilliant introduction, because what we are seeing now, or what I'll discuss, is much more practical terms, what we're doing in CoMPaSS and how we deal with the genetic data and what type of information we need so that we can proceed with the analysis that we want to do. So what I'm going to be talking about now is what we call the electronic genetic data clinical report form, and explain how we use it, how we are using it to streamline the genetic data, support the diagnosis, and also in particular enable the computational modeling for neuromuscular diseases that we want to do in CoMPaSS. So this is our brief agenda for this part of the seminar. And as you have heard many times, and you know already, one of the biggest challenges in rare diseases is what is called the diagnostic odyssey, where patients often wait years and years until they finally get an answer, genetic answer to their problems. And while doing this, patients tend to go through multiple referrals to different specialists and very often unconclusive tests. So this delay doesn't only impact patients and the families, both physically and emotionally, but also means that sometimes opportunities for trial participation or treatment can be missed. And this is why obtaining a genetic diagnosis is so important, as we all know, but not only obtaining it, but obtaining it fast. So what we want to do in CoMPaSS, our approach here is to try to standardize and digitize clinical imaging and genetic information so that we can apply computational models to help patient diagnosis. And for these computational models to be developed and tested, the project is using both large historical and also new data sets. So what is it needed to train artificial intelligence models? So we need large, high-quality data sets, both genetic and imaging. And as you've seen in the previous seminars when we discussed the MRI data. But most importantly, this data needs to be robust. And we need to be able to collect these large data sets, but we need to be able to compare them and have a homogeneous data set. So this is not a problem for the new genetic data sets that we are including in CoMPaSS. This is much easier because we have established SOPs for sample collection, sequencing, processing, all in the same way. So the different centers will get these data, these samples, and get them organized and collected similarly. But and then the sequencing will be done, as we heard from Giorgio, in CGAT. So most of the possible differences between samples, the technicalities will be eliminated. However, that's not the case for the historical data, the legacy data that we're using in the project. And this is because the historical data sets that we use in CoMPaSS come from a number of research projects that some of the CoMPaSS partners have carried out during a number of years. So these, for example, include Neuromic, CoMPaSS-NMD, and MyoSeq. These are projects that were developed and carried out in Newcastle. We've got MyoCapture done in France, INGENE2 and MyoFin also led in Finland, for example. So all these data sets, all genetic data for neuromuscular disease patients, have been somehow obtained and in by different methodologies, as we just seen in Giorgio's explanation. Once the data has been collected, it's been transferred to the Silesian University, where each of these genomic data sets will be reprocessed and used for the basis of the computational model. So this is the main reason why we need all this data homogenized somehow, because in order to be able to compare them, they need to be as similar technically as possible. And this is what the electronic genetic clinical report form provides. So it's a structured, secured way of integrating all the genetic data sets so that they can be used effectively in the computational models. So in other words, what we're doing in this form is the collection of all the relevant metadata for each patient. So first, what we have is the patient metadata. So this is the data that is specific for each patient, outside the genetic the genetics that has been done later on. So we've got the CoMPaSS ID for each patient. We've got the local identifier in order for the clinicians to be able to track them. Sex, which is the subtype of neuromuscular disease that the patient, information about the phenotype, whether the patient has an MRI, and has histopathology data also associated with this record. This is easy enough. Then what we have is all the genetic metadata, and this is what will allow us to be able to compare the data later on. So part of the genetic metadata, the first points are the type of data sets that we are using. So this could be the type of files that we'll use. So this would be FASTQ files, CRUM files, or BIOM files, depending on whether the data that we are using is data that is just out of the machine without any processing. This would be the FASTQ files, or whether the data has already some type of bioinformatic processing. So, for example, the BAMs or the CRUMs have already been aligned against a reference sequence. We then have the sequencing strategy that was used in this particular patient. And again, as Giorgio mentioned, it could be exome sequencing, it could be genome sequencing, it could be a panel, although we don't have many of these, but mostly exomes or genomes. So it will depend whether we're looking only at the exons and the coding regions, or if we're looking at all the intronic and regulatory regions as well. We need to know what sequencing kit was used. And this was, again, what Giorgio was mentioning before about how the sequencing was carried out. And he mentioned the different types of platforms that can be used. Again, every single exome, we need to have that information, because otherwise we are not able to compare them. Different sequencing kits and different will have different target regions, and depending on the target regions, if different different kits are used, we might not be able to compare the same genes if in one of the kits some genes are covered and not in the other one. So it's very important to know what's the target that we're looking at. The reference build, this is how the data has been processed. So in the case of data sets that were not straight off the machine, but they were aligned against the reference, we need to know which reference build has been used. So we've got different genome references. As we move on, a number of years ago, we were using build 18, then we moved to build 19, and now we're in build 38. So it's important to know which one has been used to process that data set. Otherwise, again, it won't be comparable. What machine has been used for the sequencing, and also who was the sequencing provider. Again, maybe not as relevant as some other aspects of the technicality, but it's also important to know where the data has been, the sequencing has been done. And then last, information about the batch, or when and if all the samples were sequenced together, if it were different batches, if there were any information that we've got about when the data was obtained. So all this information constitute what we call the electronic genetic data clinical report form. And once all the metadata is accounted for, the genetic data can now be processed in a common standardized bioinformatics pipeline. So I mentioned before that all these data sets were transferred to the group in Poland that are doing the processing of the data. And this processing in our case is based on GITK best practices, again, to make sure the data remains robust and comparable. And once that is done, then what we can do now is utilize this data for the computational models. So in summary, the EGCRF is the collection of metadata that within CoMPaSS-NMD will help us to capture, standardize, and integrate the genetic information in our neuromuscular disease patients. It provides the foundation for the computational modeling within CoMPaSS, and it will help us in the future diagnostics that will come from the results that we get in the project. So that's the overview of how we implement and how we deal with the large data sets that we are collecting and how we move forward within the project. Thank you very much for your attention, and if there are any questions about this, I'm happy to discuss them later. Thank you.
Thank you, Anna. Very, very nice presentation, very detailed point of view. Yes, here also your contact in case somebody want to ask you remotely. Thank you for this presentation. So I guess we can move to the next section. Is a section of questions and answer. I invite people in the audience to make questions. Anna will reply to your question related to the ECRF. She has details on genetics, but perhaps she can help me and Rosella to try to answer also to issues raised by the previous Giorgio's presentation. Are there questions to Anna? I cannot see in the chart, but Giselle Bonnet. Giselle, please.
Yeah, many thanks, Anna. It's very, very nice work and very interesting development. I was wondering at some point, as I already highlighted at the previous meeting, not during a webinar, but previous meeting, that there is obvious link or link between different EU projects, this CoMPaSS-NMD and what is currently developed within the European Research Alliance for Rare Diseases, whereas we are going to reanalyze, as we did in the previous EU program Solverdi, some genetic data in coherence with the phenotypic data. And so what is built within CoMPaSS for the neuromuscular field is very interesting because it's integrated and very comprehensive metadata from clinic to genetics that would be perfectly fitting with the pipelines that are under development for RDRA. So at least for neuromuscular diseases, I think obvious link, and I was wondering whether within CoMPaSS, and you had already thought this, how to liaise the two projects.
I can
Thank you.
answer because Giselle, I applied, but I was not included in the panel. So probably we can organize and try to integrate the atlas with what Erdera is building. Of course, we have to find a way.
Mm-hmm.
Because as you suggested, I applied, but I was not included.
So you applied
As
for...
to be a member, this was last year, when
Yeah.
there was the call for the panel and this, and I applied, but I was not included.
Okay, but there might be next step, and I think Anna, if I'm not mistaken, you have a role in the super DIFT group. So
Correct.
maybe it's another way. There is official integration, but there is, start to demonstrate the added value.
Yeah.
And with this type of demonstration, then reapply, because I'm sure there will be several rounds of application to join as beneficiary the project.
Yeah, one of the things that we did with Joanna Polanska, who is leading the computational model, was to access the previous Solvar-D data.
Mhm.
So what we wanted to do was to use the Solvar-D data as part of the training data sets.
Mhm.
And that would be perfect, because if using that data, we could show that there is some structuring and clustering, and then feedback, as Rosella was saying, and in the second application, maybe show that there are some results, then
Yeah.
that would, and also it doesn't need to be only for NMD. Of course, this is,
No, it
you know,
can expand
it could
after
be, it could be
any indication.
exactly.
Yeah, I think it's a very nice use case as a
Yeah, exactly.
demonstrator, and then it's a win-win situation both for CoMPaSS and for Erdera. And
Yeah, I have another comment, because yesterday I met Marco Savarese, because we were talking about the data, probably our collection with the retrospective data, and the other data will be a very large collection of data on neuromuscular patients. So definitely it is something to pursue.
yeah, I remember after the Strasbourg meeting, I was in connection with the coordination team, and I liked CoMPaSS and all the added value of it. So we can keep contact and advertise CoMPaSS again and again, and at some point connection will be officially done, and I know that all the coordination and administrative things are very cumbersome, and the coordination team has been a bit under the water, understaffed recently, and that may be one of the reasons why your application, Rosella, was not included yet. But I'm confident that in the future, at some point, there is no way but being part of it.
Yeah.
Yeah,
At least we are several already in, and we will push hard for that.
I mean,
I'm
many of
sure
us,
Anna,
exactly.
Volker,
Many of us are
Marco,
in
we will
both.
all
Yeah.
do all our strong lobbying to take advantage of all what you are developing.
Okay.
Great. Are there other questions? I was wondering, Anna, if in the large set of training data, and also with the plan to include the Solvar-D data, if anybody has thought to pursue possibility to include a large set of 100K GP data. I mean, I'm reading and reviewing personally a number of papers now going to several high top journal using the data, and I was wondering if is there a way to explore also this large collection of information?
Yeah. So the problem with the 100K genome data is that you can apply to look at the data in situ, but you cannot download the data. So they've got a very strict way of accessing the raw information. So you need first of all, you need to be approved and all the rest, but also then to be able to work on it, you need to log into their system and do all the analysis in the cloud, let's say. But it's not allowed to retrieve raw data for something like what we can do. At least that's my experience. I don't know if I mean that's what the working level, but maybe at a higher level it is possible to apply that. Maybe it's something that Rosella could.
You mean even if you are based in the UK, you can?
Yeah, it's not related to the location. It's just that all the data is within the same cluster and the same cloud and cannot be downloaded.
Okay.
Yeah, it's a shame really, because it's a huge amount of data that would be,
it's like the large data sets
exactly.
in the US, exactly the same.
Yeah.
Well, other questions? If not, I thank you all. I just a reminder, we will let you know the exact date for the next seminar that is really attractive on the Atlas tool, that is the blueprint of this project. And so please don't miss the next junior investigator training webinar in the spring. And with that, I thank you all, and I wish you will join us for the next webinar. Thank you very much.
Bye.
Thank you. Bye.
Bye-bye. Thank you.
Bye.
Cheers.